Before starting any of your processes, you need to ensure that you've undertaken hand decontamination through hand washing, and that you're wearing all the required personal protective equipment that is appropriate for the task you're about to undertake. These will be listed in your standard operating procedures and will include items like lab coats, gloves, goggles, and masks as needed. Once you've finished with the hand wash and put on your PPE, you'll need to prepare your work areas. This includes taking out any of the consumables that will need to be brought to room temperature, as well as cleaning down your workspace. You'll also need to do things like prepare disc pots that contain 10% bleach. Your workspace must be cleaned at the start and end of every day, as well as in between changes of procedure when you'll be working with different specimen types. The same process will also need to be undertaken if there are any spillages. For fluid spillages, you'll need to follow local guidelines, as there may be additional steps that you're required to undertake. Preparing and cleaning down of your workbench includes not just cleaning down the bench space itself, but any of the equipment you're likely to use, including racks and pipettes. We're now going to look at how to read an e-test. Record the temperature of the incubator when you remove your e-test plates, as well as ensuring they've been in the right kind of incubator, i.e. CO2 for Streptococcus pneumoniae. Ensure that you always read the matching control plates at the same time as the specimen test plates, and when reading the plates, the growth should be evenly distributed across the plate and the inhibition zones should be uniformly visible. Individual test samples should be compared at reading with the density of the control plates. If the density of the test sample is greater than that of the control plate, the test should be repeated as isolates may appear to be falsely resistant. If individual colonies can be seen on the bacterial lawn, the inoculum is too light and the test should be repeated and not read. Do not read the plate if the culture appears mixed, but produce a purity plate for each morphology or resistance type, then reset up antibiotic sensitivity plates and follow up with an e-test as required. MIC breakpoints are usually clear-cut, but different growth inhibition patterns may be seen. Read the MIC value at the point where no growth is visible in the gradient. If the inhibition zone is asymmetrical, then read the MIC at the higher value where the growth meets the e-test strip. If the growth intercepts between two values on the e-test strip, then the MIC is recorded as the value of the lower section. We're going to cover 4 types of e-test where you may need to repeat. The first is mixed, and so you'll require a purity plate and to repeat both the antibiotic sensitivity testing and the e-test. The next version is a bad 3-way streak, so where you haven't inoculated the bacterial lawn evenly. This will require you to repeat the e-test itself. The third is a mixed plate with mixed resistance, so you have one resistant and one less resistant set of bacterial isolates on the plate. This results in a double zone. You need to treat this as mixed, and so separate the two and then repeat both the antibiotic sensitivity testing and the e-test if required. Finally, this is a badly spread bacterial lawn using a rotary plate. You probably won't be using rotary plates to set up, but it gives an example of how, if you don't actually swab out evenly, you can get differential spread across the plate, which means that the test is actually invalid and you'll need to start again. You will then need to clean down your bench space appropriately.